Hello everyone. So today we begins with the physiology of gastrointestinal tract, and in this video we are going to cover the biliary system. So the biliary system includes the liver, the gallbladder and associated ducts. These include the right and left hepatic duct, common hepatic duct, cystic duct, common bile duct. So this is the biliary system, which is made up of liver, the gallbladder and this type of hepatic duct, cystic duct and common bile duct. So this is the liver, which is the most important part of the biliary system. So these are the two hepatic duct, right and left hepatic duct, which is joined to form the common hepatic duct. This is the cystic duct, this is the gallbladder, and gallbladder opens into the common hepatic duct, and now this is known as common bile duct. Common bile duct joins with the pancreatic duct and it opens in the duodenum, which is the first part of the small intestine. Alright. Now the functions of the liver. Most important Viva question. Which are the functions of the liver? Synthesis and secretion of bile, metabolic process which includes gluconeogenesis and glycogenolysis. Here there is a detoxification and degradation of different type of drugs and hormones occurs inside the liver. Synthesis of plasma proteins, for example albumin and clotting factors. Storage of the iron and vitamin B12. Activation of vitamin D. Removal of bacteria and old RBC, excretion of cholesterol and bilirubin. So all these are the functions of the liver, and this is the most important Viva question. Which are the functions of the liver? Synthesis and secretion of bile, metabolic process which includes gluconeogenesis and glycogenolysis, detoxification and degradation of different type of drugs and hormones, synthesis of plasma proteins, for example albumin and clotting factors, storage of iron and vitamin B12, activation of vitamin D, removal of bacteria and old RBC, and excretion of cholesterol and bilirubin. So all these are the functions of the liver. Now the bile. What is the characteristic as well as the quantity of the bile? It's a green viscous liquid around 600 to 900 ml per day, having the alkaline pH so that it is going to neutralize the acidic chyme. It is secreted by the liver via bile duct to the duodenum, and once it is formed inside the liver, it is stored inside the gallbladder. The storage of the bile occurs in the gallbladder when it is not required. The functions of the bile is the emulsification of the fats, as it helps in the digestion and absorption of fats. Now the composition of the bile, which includes bile salts. These are the salts of glycolic and tarcholic acids, bile pigments which includes the bilirubin and biliverdin, and cholesterol. This is the composition of the bile. 95% bile is reabsorbed and returned to the liver. So whatever the bile, once it is released into the small intestine, again it is reabsorbed, and via the portal circulation it again enter into the liver. Inside the bile there is not any type of enzyme is present. There isn't presence of any type of enzyme. So that is the important feature of the bile. Bile only have the bile salts, bile pigments and cholesterol. Bile salts, these are the glycolic and tarcholic acid salts. Bile pigments includes the bilirubin and biliverdin, and another composition, it is the cholesterol. It is secreted by the liver and it is stored inside the gallbladder. When it is required, it is released into the small intestine. Bile salts. the most important component of the bile. They are sodium and potassium salts of bile acids. These are the sodium and potassium salts of glycolic acid and tarcholic acids. They are derivatives of cholesterol and recycled through the enterohepatic circulation. This is what about the bile salts. It is the most important component of the bile. They are sodium and potassium salts of the bile acid. For example, sodium glycolate, sodium tarcholate, potassium glycolate, potassium tarcholate, etc. They are derivatives of the cholesterol and recycled through the enterohepatic circulation. Inside the liver, bile salts are formed from the cholesterol. When there is a need of the bile salts after the heavy fat diet, it is directly enter into the duodenum, which is the first part of the small intestine. But when there is no need of the bile salts, it is stored inside the gallbladder. When there is a need, the gallbladder will contract and release its bile along with the bile salts into the first part of the small intestine, which is the duodenum. Now this bile and bile salts, all the things travel to the jejunum, and at the end of the ileum or the terminal ileum, this bile and bile salts are reabsorbed, and via the portal circulation it again enter into the liver. So this is known as enterohepatic circulation of the bile and bile salts. Function of bile salts: emulsify large fat particles into smaller ones that can be attacked by the lipase, so having the detergent action, help in the transport and absorption of fat, having the micelle formation. It prevents precipitation of cholesterol by keeping them into the solution, so it prevents the gallstones and stimulate bile secretion by the liver cells. All these are the important functions of the bile salts: emulsify large fat particles into smaller ones that can be attacked by the lipase, so it having the detergent action, helps in the transport and absorption of fat, having the micelle formation. It prevents the precipitation of cholesterol by keeping them into the solution, so it prevents the gallstones. It stimulates bile secretion by the liver cells. So these all are the functions of the bile salts. Composition of the human bile. Now whenever the bile is synthesized inside the liver, the concentration of the bile, which is at the time of synthesis, and when it is stored inside the gallbladder, having the much difference. So here you are saying that the water content inside the hepatic, it is 98%, but inside the gallbladder it is only 89%. So inside the bile salts 1.1 and inside the gallbladder 6. Bilirubin 0.04 and inside the gallbladder 0.3. Cholesterol 0.1 in the hepatic and 0.3 to 0.9 in the gallbladder. Fatty acid 0.12 inside the hepatic and 0.3 to 1.2 inside the gallbladder. Lecithin 0.04 in the hepatic and inside the gallbladder it is around 0.3. So electrolyte content of the bile, which is also difference between the hepatic and gallbladder. So here the inside the hepatic there is a higher amount of the sodium ion is present, whereas inside the gallbladder there is a higher amount of the calcium ion, potassium ion and chloride ion, which is present inside the hepatic, which is in higher amount or in the higher concentration. So that is the difference between the electrolyte content of the bile in the hepatic and inside the gallbladder. Functions of the bile, mostly due to the bile salts, obviously. Absorption of fats, it having the emulsification of fats and having the detergent action. Micelle formation and having the Fary action. Absorption of fat soluble vitamins. Activation of pancreatic lipase. So this helps in the absorption of fats, and it is the important functions of the bile and mostly due to the bile salt. Cholerectic action, there is a two words, cholerectic and cholegogue. Cholerectic action means stimulation of bile secretion, and cholagogue action means contraction of gallbladders. These two are the different words. Cholerectic means stimulation of the bile secretion, where cholagogue means contraction of the gallbladder. So there are certain substances which are some of the cholerectic and some of the cholagogue. So the cholerectic substances, it stimulates the bile secretion, where the cholagogue substances, it causes the contraction of the gallbladders. It keeps the cholesterol in the solution in the bile. Mild cathartic action, or simply it means increase the frequency of the stool, or anti-constipative action. Excretion of cholesterol, drugs, toxins, bile pigments, inorganic substances. So there will be also along with the bile many type of cholesterol, drugs, toxins, bile pigments and inorganic substances are excreted inside the gastrointestinal tract. Neutralization of the acidic chyme. So there will be the rich in bicarbonate ion inside the bile. So there will be the neutralization of the acidic chyme. Inhibit endogenous synthesis of cholesterol by the liver. So these all are the functions of the bile, and the functions of the bile is most important due to the bile salts. Absorption of the fats, cholerectic action which means stimulation of the bile secretion, cholagogue action means contraction of the gallbladder. So these three are the important functions of the bile. Regulation of bile secretion and gallbladder emptying. How the bile secretion is regulated and how the emptying of the gallbladder is also regulated. So chemical. First of all, chemical mechanism. Bile salts, most important stimulus of the bile secretion by the liver cells. Bile salts are the most important stimulus for the bile secretion by the liver cells. So here is the importance of the bile salt. Hormonal regulation, there is a secretin and cholecystokinin. The whole of the GIT is mainly dependent upon the three hormones, gastrin, secretin and cholecystokinin. Everywhere these three hormones, but mainly secretin and cholecystokinin is present. So secretin, it is secreted in response to the acidic chyme. It causes secretion of bile rich in water and bicarbonate ion. Cholecystokinin, secreted in response to fatty acids in the duodenum. It causes gallbladder to contract and sphincter of Oddi to relax, so that the bile is released inside the duodenum. So this is the chemical regulation of the bile secretion. This is the hormonal regulation of the bile secretion. And now the next one, it is the nervous regulation or the neural regulation of the bile secretion via the vagal nerve or via the vagal stimulation. It increases bile secretion and there will be the weak contraction of the gallbladder. So this is the vagus nerve. Whenever the stimulation of the vagus nerve, it causes the contraction of the gallbladder via the acetylcholine, and so the bile is released into the duodenum. Whenever there is a presence of fat particles inside the small intestine, this stimulates the eye cell of the gastric mucosa to release the cholecystokinin. And cholecystokinin also causes the contraction of the gallbladder, so that the bile is directly released into the first part of the small intestine, which is the duodenum. The three mechanisms of the secretion as well as the gallbladder emptying: chemical mechanism, hormonal mechanism and neural mechanism. Jaundice, which is the applied aspect of this biliary system. What is jaundice? It is the yellowish discoloration of the skin, sclera and mucous membrane, which is due to increased blood bilirubin level. So there are three types of jaundice: pre-hepatic jaundice, hepatic jaundice and post-hepatic jaundice. Pre-hepatic jaundice due to excess production of bilirubin, for example hemolytic anemia. Hepatic jaundice, in the liver disease, there will be the conditions of hepatitis. And the post-hepatic jaundice, whenever there is obstruction to the bile flow, either due to the gallstone or any other type of obstruction to the bile flow, there will be the jaundice. So what is jaundice? It is the yellowish discoloration of the skin, sclera and mucous membrane due to increased blood bilirubin level. Now the liver function test. Liver function test will be asked as a short note. And there is another one short note which I already told you about the biliary system. So these two short notes are most important. First one liver function test and second one the biliary system. Test to assess the secretory functions of the liver. So which are the tests we have to perform for the assessment of secretory functions of the liver? Serum bilirubin, urine bilirubin and bile salts, urine urobilinogen, fecal stercobilinogen and fecal fat level. So all these are the tests to assess the secretory functions of the liver. Serum bilirubin, urine bilirubin and bile salts, urine urobilinogen, fecal stercobilin and fecal fat level. Now the test to assess the metabolic functions of the liver. This includes the galactose tolerance test, blood glucose level, blood and urine amino acid level and lipid profile. So all these are the tests to assess the metabolic functions of the liver, which include galactose tolerance test, blood glucose level, blood and urine amino acids level, lipid profile. Third one, test to assess synthetic functions of the liver. So how we are going to assess the synthetic functions of the liver? So we have to test the estimation of the plasma proteins, serum levels of the liver enzymes, for example SGPT, SGOT and alkaline phosphatase. Here SGPT means serum glutamic pyruvic transaminase and SGOT means serum glutamic oxalate transaminase. So SGPT means serum glutamic pyruvic transaminase, SGOT means serum glutamic oxalate transaminases. So all these are the liver enzyme we have to assess for the synthesis functions of the liver. Next one, blood urea, blood ammonia, urine ammonia and coagulation factors. So all these are the tests to assess the synthesis functions of the liver, which includes the estimation of the plasma proteins, serum level of liver enzymes, SGPT, SGOT and alkaline phosphatases, blood urea, blood ammonia, urine ammonia and coagulation factors. Fourth one, test to assess hepatocellular integrity. How we are going to assess the hepatocellular integrity? So this is the ultrasonography, CT scan and liver biopsy. We have to do the ultrasonography or the USG of the liver, CT scan of the liver and liver biopsy to assess the hepatocellular integrity. Fifth one, test to assess detoxification functions of the liver. So here we have to do the bromsulfathalin excretion test. How we are going to assess the detoxification functions of the liver? So we have to do the bromsulfathalin excretion test. I hope in this video or via this video you are going to understand about the biliary system as well as the liver function test.